Now I would like to present our first presenter, Dan Itzhak. He's a postdoctoral fellow in Systems Biology of Membrane Trafficking Group at Max Planck Institute of Biochemistry in Martinsried. He obtained his bachelor's degree in natural sciences at the University of Cambridge before moving to the Institute of Cancer Research in London to complete his PhD in mechanism-based drug discovery. He moved to Max Planck Institute of Biochemistry to work with Dr. Georg Borner in developing a method for comparative spatial proteomics. The method is capable of detecting protein subcellular localization changes on proteome-wide level. And he will talk on MS-based proteomics today.
Okay, good morning, everyone. So I just want to start with a show of hands for how many of you in the audience have access to an Orbitrap-based mass spectrometer? Fantastic. And anyone who does not, actually? Okay, one. It was the same as last year. Okay, so as you can see, the title is MS-based proteomics. And essentially, over the next 40 to 45 minutes, I'm going to discuss a lot of the different technologies and hopefully get across to you why I believe that the development of the Orbitrap-based mass spectrometer is a greater technological achievement for mankind than is putting a man on the moon. So today, the technology is so advanced that the mass spectrometer instrumentation is no longer the limiting factor. You are. So what we can do with it, with mass spectrometry now, we can measure the abundance of proteins in different samples, whether that be sample treated with different drugs or different patients. We can get deep enough that we can measure all the different isoforms that are present. We can look at the protein distribution in different organs and also in different body fluids, so plasma and CSF, something that is not yet achievable with other omics technologies. We can look at the turnover of proteins in the cell. We can look at how they change with circadian rhythm or during other time courses. And now even single-cell proteomics is within reach. We can look at post-translational modifications. Example is phosphorylation, but there's almost no limit to the different modifications that you can look at with mass spec. You can look at interactions, subcellular localization, protein stability, conformation, and even structure, all with a single mass spec instrument. So how does the workflow begin? We take our cells or tissue or body fluid, extract proteins, convert them into peptides with digestion, and use HPLC typically to separate our peptides to make the mixture less complex, inject those into a mass spectrometer using electrospray ionization, and then operate the mass spectrometer with cycles of full mass spec followed by MS-MS for sequencing. So you get your full MS for quantification and your MS-MS for identification, typically. So let's just have a quick look at the architecture of a mass spectrometer. So this is probably architecturally the simplest mass spec, or one of the simplest mass spectrometers, the QExactive. It's also one of the best performing mass spectrometers. So we start in the bottom right-hand corner. This is meant to symbolize an electrospray. So the ions are converted from the liquid phase to the gas phase via this electrospray, and they enter into the mass spec and are focused through the S lens, and this they pass around a bend called the injection flatter pole. This is where non-charged ions don't make it around the corner, basically, and then they're filtered on a quadrupole, and they can be filtered by their mass-to-charge ratio by altering the radio frequency, which is applied to the two rods, to the two sets of rods here, and then the ions pass they're accumulated in the CTAP and injected as a packet into the Orbitrap where they're measured. Now, I'm not going to do an injustice. I'm going to let Professor Makarov describe exactly how this machine works. But suffice to say that we get our MS1 scan by injection into the Orbitrap from the CTAP, and then when we want to do our fragmentation, we put the ions into this HCD cell where our ions collide with gas and fragment, and then they go back into the CTAP and then are injected to get the fragmentation patterns. So just typically, this is operated in the following way, where you allow accumulation of ions by setting the quadrupole, and then you acquire a high resolution scan for the full MS, followed by n number of data-dependent or potentially data-independent MS2 scans for fragmentation. So what do I mean by data-dependent and data-independent? So in data-independent mode, or data-dependent mode, sorry, as our species, our ion species, elute from the HPLC and are detected by the mass spectrometer, we select the top n most abundant species for fragmentation. And in that way, we get the identification of the things that we can actually see in the MS1 scan. And that occurs semi-stochastically over the course of the HPLC run. Now, in data-independent acquisition, you ignore you ignore the data in the sense that you ignore the intensity of the precursors that you can see in the MS1 scan, and you just fragment everything in a window, and you get very complicated MS2 spectra. But you also don't lose or omit any of the ions that are eluting at any one time. So the data-independent acquisition, for that strategy, you really need different software. And for that, I can recommend any of these different softwares. And this paper by Navarro in 2016 in Nature Biotech is a good source for you to begin. There are also summer schools that you can attend for all of these different softwares. But the most important thing I would say is that for all of them, you still need a data-dependent acquisition spectral library in order for this to work. And for that, I would recommend coming here. So we're not going to talk about data-independent acquisition for the rest of the talk. We're going to talk about data-dependent. And I'm going to take you through a lot of the popular quantitative proteomic strategies. So we're going to start with MS1 labeling strategies, move on to MS2, and finish with label-free. So essentially, this is SILAC, TMT, LFQ. Those are the main things that we're going to be discussing. This is essentially the strategies that are used in order to make mass spec quantitative, because it's not inherently quantitative, right? The peptides that we inject into the mass spec have different physicochemical properties, and it means that peptide X cannot be directly compared with peptide Y. So in MS1 labeling strategies, in fact, all labels are isotope-based. There isn't a single label that is not. So we use actually stable forms of stable heavy isotopes, and these do have the same physicochemical properties, but they have different atomic masses. The exception is deuterium, but we'll ignore that for the time being, and just say that when we incorporate these heavy isotopes into proteins, it enables the mass spec to distinguish the resulting peptide species. Okay, so how do we get these heavy isotopes into cells? And that's what this MS1 labeling is. So it actually occurs in the living organism. And this is, to some extent, a limitation of MS1 labeling. You have to begin by labeling your species with these heavy isotopes. The beauty of this approach is that you overcome systematic errors that come about during digestion and peptide fractionation, and also every single thing that happens to your peptide when it's eluting from the HPLC all the way to the Orbitrap where it's measured is the same. There's nothing that you need to control for there. Okay, so let's have a look at the most popular method, which is SILAC, which stands for Stable Isotope Labeling by Amino Acids in Cell Culture. So you have your two different cell types, and you already have to start growing them apart, let's say, or if you want to treat this one with a different condition, you're going to need to grow them separately. So we use actually arginine 10 and lysine 8, where in each case, all of the carbons and all of the nitrogens are heavy isotopes. So you have this mass difference of 10 Daltons for the arginine and 8 Daltons for the lysine. And the cool thing about this is that both of them are essential amino acids, in that the cells, or mammalian cells at least, will not be able to survive in the absence of supplemented arginine and lysine. They won't be able to synthesize their own. So when you feed it to them, when you feed them only heavy arginine and lysine, they will become incorporated into all of your proteins. And if you wait for 7 doublings of the cell, only 1/128th of the material will be light labeled afterwards. So then we extract our proteins. And the next cool thing about this is that we use trypsin for cleavage, which cleaves exclusively C-terminal to arginine and lysine residues, which ensures that every tryptic peptide has a precise mass shift. So we know when we see a peak in the mass spec that belongs to a peptide that you're either looking at the light or the heavy form, and then you can look 4 Daltons upstream or 4 M over Z upstream or downstream to find the Lys-C labeled peptide. So then when we look in the MS1, what happens is that the ratio of the light labeled peptide to the heavy labeled peptide is indicative of the protein abundance from which those peptides were derived. So if you see a different ratio, you know that it was differentially expressed in that cell type. Okay, so one of the limitations of SILAC is that it's actually only capable of multiplexing to a maximum of 3 channels, really. And another thing is that each channel increases the spectral complexity and reduces the depth that we're able to achieve. So by that I mean that if you imagine if you have a single channel, if you have only light labeled peptides, then the top n most abundant peaks in your MS1 scan are all going to belong to different peptides. But if you have now a second channel or a third channel from the heavy labels, then the top n most abundant peaks may not actually belong to different peptide species, but they could well belong to the same one. So the sequencing depth that you achieve is lower. So I already mentioned that you can get 3 channels. You can get the third channel by using lysine 4 and arginine 6. And you can see that the isotopic pattern that it gives rise to is in between the fully heavy isotopic labeled peptide and the light labeled peptide. So you can see that from this, that if you try to add in another channel, it would start to overlap with the ones that we detect with the 3 we already have. And if you want to get higher multiplexing, if you want to kind of retain this sensitivity of SILAC-based proteomics, you can use a SILAC spike-in, where you spike one of the standards into every experiment that you're running. So in this case, this medium-labeled condition is spiked into one experiment with a heavy and a light, and then a second experiment, and a third, and a fourth. And by that, you're able to keep this high accuracy quantification across. So there have been some recent technological developments to try to overcome this limitation of SILAC. So this was the explanation of the reduced depth. I already went over that. And this was from Josh Kuhn's group, who's going to be here later in the week. And it's called NuCode SILAC, neutron-encoded SILAC. And this takes advantage of the fact that you can label your lysine with 8 heavy isotopes. And if you use those 8 heavy isotopes from different elements, they have a slightly different mass. So if you have just 6 carbons and 2 nitrogens, that's slightly different from having 3 carbons, 4 hydrogens, and 1 nitrogen. There's a I think it's 36 or 18 millidalton difference between these two forms of lysine. Now, to take advantage of this, if you scan these isotopes together in a low resolution scan, in the MS1, you cannot resolve those two peaks. Only when you use a very high resolution MS1 scan do you actually see that these two peaks begin to separate. So your quantification from new code SILAC comes from looking at the ratio between these pairs here, and then your low resolution scan just tells you which peptides you should take for MS2 sequencing. Okay, so as I mentioned, that one of the interesting things that you can do with SILAC is look at things like protein turnover. So this is a nice paper from Matthias Selbach's group published in Cell in 2016. And what they've done here is they have 3 labeled cells, heavy, medium, and light, and they pulse them with azidohomoalanine, which is a replacement for methionine, and it will be incorporated into every new protein. And then you chase it. So it's a typical pulse chase experiment. So I'm sure those of you that have done biochemistry will have seen this done with radioisotopes, where you actually read out the pulse chase via X-ray film. And in this case, we're chasing it with cold amino acids that do not contain heavy isotopes. And then we can combine all of the samples and use click chemistry to enrich for these newly labeled proteins, and we can differentiate them because they have different masses, because they contain different SILAC amino acids, and now we can quantify them. So they use this to look at how proteins degrade with time. So here we can look at a protein which is really stable. So at time 0, we can see that there's good incorporation. This is the heavy label, and then after 1 hour with the cold chase, we still see that most of it is left, and still after 2 hours as well. Unlike this protein here, basogin, which is rapidly degraded after 1 hour and then becomes stabilized. And this is a really nice paper, and I encourage you to read it, especially if you're interested in protein turnover. Another thing that we can do, and this is really powerful, is we can actually culture 2 cells or 2 cell types together and get one of them to become SILAC light labeled and the other to become SILAC heavy labeled. And why that's really useful is that you could then kind of put this mixture of proteins from 2 different cell types into the mass spec, and you know which proteins come from which cell type. So how is that achieved? Well, we can, as I told you before, if you grow cells without lysine, or if you don't supplement lysine, they don't grow. So you need to add it back, and that's how standard SILAC works. But with this CTAP, this cell type specific labeling, what we do is we provide a precursor of lysine, and we modify genetically that cell line with an enzyme which can convert that precursor. So by coupling these 2 together, one cell type will be labeled with a lysine. And the second cell type, you give it a different enzyme and a different precursor, and then this can be labeled with lysine. And you just make one of the precursors heavy and one of the precursors light. And now we can separate them by mass spec and see which proteins came from which cell type. Just one thing, it's a small note, is that not everything can be SILAC labeled. So, for example, plants, they metabolize lysine, and essentially the heavy label that you put in there gets put into other amino acids, like proline. And that complicates that complicates the analysis. So it is possible to label just with nitrogen 15, but this is not so great because the number of nitrogen 15 labels varies from peptide to peptide, leading to an unpredictable mass shift and complicating the data analysis. And several people have asked Jürgen in the past, why is this not supported in MaxQuant? Well, because it's like very challenging, and there's plenty of good alternatives available in case you want to do this. So let's just take a quick summary then of the metabolic labeling approach we've just looked at. So the strengths are that they overcome systematic errors. They're very accurate. It's quite simple to achieve, and it offers powerful experimental opportunities. On the downsides, though, it leads to this increased MS complexity, which reduces depth. It's only really suited to organisms which are dividing. There are some methods around that, like the super SILAC approach. And there's limited multiplexing. And there's also a limited dynamic range of the ratios that you can achieve at MS1 level. Okay, so now we're going to look at chemical labeling. And one of the reasons that kind of spurred the development of chemical labeling is the lack of multiplexing that's achievable by MS1 labeling. So in this case, the labeling occurs after the digestion into peptides. It's done chemically, by chemical reactions. So this allows greater multiplexing, and the interesting thing here is that all the peptides are isobaric in MS1 and give rise to reporter ions in MS2 or MS3. And this means that our MS1 spectra are not any more complex, and the MS2 or MS3 spectra give us the 6 different channels back again. So how is that achieved? I'm going to talk about, as I said, tandem mass tags as the kind of the prototype for all of these isobaric approaches. There's also something called Itzhak, which you can read from these references here. But essentially, the TMT reagent looks like this. You have a NH2 reactive group. This N-hydroxycinamide ester group will react with free N-termini and free lysines in your peptide. And this then couples this other part of the molecule, which contains a mass normalizer and a mass reporter. And again, I told you, right, that we always use heavy isotopes when we want to separate things by mass spec. You can have 6 different versions of the tag, and so when you, I forgot to mention, when you fragment this reporter with high-energy collision-induced association, the peptide, this reporter will break at this bond here. So in this tag, we have 5 heavy amino acids. 5 heavy isotopes on this balancing part of the reporter, and this has none. So when this breaks apart, this will have a mass of 126. But if you now put 1 heavy isotope there, it will have a 1 Dalton mass shift, and so on until you get all of these 6 channels. So they also took advantage of this very small mass difference between different heavy isotopes, as in there's basically a 6 millidalton difference between something which contains a heavy carbon and that which contains a heavy nitrogen on the same molecule. So here we can see that this one contains the heavy nitrogen. It's 127.12476 daltons, and then the one with the heavy carbon is 127.13108. And the mass spec can really detect this difference, so it allows you to have 5 extra channels. So each of these channels here, you can use this trick to give you now 11. There is one downside to this approach, to the TMT approach, and it's the ratio compression problem. It's the fact that when we try to isolate our peptide using the quadrupole, instead of isolating only the target ion that we're interested in, we also isolate near isobaric ions. And this gives rise to, so it's when you measure the reporters from this, you will detect the reporters not only from this peptide, but also from this peptide. and that's termed ratio compression. So if you take yeast at a ratio 10 to 4 to 1, 1 to 4 to 10, and spike in a human cell line at 1 to 1 to 1 or nothing, then you can look at the interference that comes from having this extra spectral complexity from the human, and all of these will give rise to reporter ions. So essentially what happens is where we expect to see a ratio of 10 to 1 here, when we have our human peptides as part of the thing as well, which they're also labeled, right? You actually only see a ratio of 3 to 1 because of this interference. So there is also a beautiful solution to that, and it's called synchronous precursor selection MS to the third, and it's done on a fusion. And if you have a fusion, I recommend that you try this because it's actually really beautiful to look at. So how does it work? Let's go through the process. So we start with a full scan on this is the architecture of an Orbitrap Lumos fusion instrument. So this is our full scan. These are all of our different peptides, and we inject those into the Orbitrap, and we get our MS1 scan. And we pick. Remember, we start with the top N most abundant features. So we pick this most abundant peptide for sequencing. So we isolate the peptide with the quadrupole, and you can see already that we've co-isolated a near isobaric peptide. This is injected into this dual cell linear ion trap, which is a second instrument that's on the Lumos that's available for MS2 or MS to the N capability. So we look at our MS2. We see we've actually told you we've isolated some of this co-eluting peptide. So we fragment it in the high pressure part of the linear ion trap, and then we look at it in the low pressure part. These are ion multipliers here, and we get this MS2 collision-induced association spectra. And now what we're looking at is fragment ions that match to the target peptide, and in gray we have unmatched fragment ions. So these are the ones that belong to our co-eluting peptide. And the idea with this is that the fragments that match to the target peptide are likely to be more abundant, and if we just take the most abundant ones, then we're going to avoid this co-eluting fragments. So what do we do with the mass spec? We actually isolate these ions again. This time we put them into the high pressure cell, fragment them, and we're going to apply a waveform to eject all of the ions that we don't want, right? So we go to that spectra which contains those fragment ions, those abundant fragment ions, and we kick everything else out using this exciting everything that's not on that inclusion list. So instead of measuring this, what we're going to do, we're going to fragment it again at really high energy and inject those fragments into the Orbitrap for an ultra-high resolution analysis. And you need that ultra-high resolution for this TMT 10 or 11-plex so that you can see this 6 millidalton difference between some of the channels. So now we can see these 10 report ions, right? So there are two that are very close together. This is the 6 millidalton difference, and then these two are 1 dalton away from each other. So that's how we, okay, and just to say that in parallel, whilst it's doing this ultra-high resolution scan, the machine doesn't sit idle. It starts to accumulate the next ion, and it does this MS2 scan. So this is quite slow. So I think currently it's capable of doing like top 5 or top 10. And there are alternative solutions. And as you will have seen from in Matthias's talk yesterday, there's this newly developed tags, easily abstractable sulfoxide isobaric tag. So you can see that in architecture, it's actually quite similar to TMT, where you have this reactive group that will, again, couple to free N-termini and free lysines, an equalizer group, and also this MS cleavable site, and then a neutral loss group. And in this case, instead of cleaving away cleaving off the reporters and reading out the intensity of the reporters to quantify our different samples, what we're doing is actually looking at the peptide-coupled reporter ions. So in this case, we have yeast peptides spiked in 10 to 3 to 1, so in black, red, and blue, respectively. And you can see this is a human peptide here in gray, and there's absolutely no interference with the ratios, right? You can distinguish these peaks from each other, and you know that this does not belong to the yeast peptide, and you don't include it, right? So whereas if this were the MS3 or rather the TMT approach, these would contribute to your ion signal. So this comes with it comes with this thing that you have to do to select the monoisotopic precursor when you do this. So you have to isolate only the carbon-12 peak. Otherwise, these peaks start to merge into one another that you get from the second and the third isotopic pattern. Okay, so isobaric labeling then. The strengths are that you have higher multiplexing for increased throughput. There's no increased complexity at MS level. You have very high precision when you measure these, just like you would in SILAC, and there are fewer missing values within one run relative to a label-free approach. The weaknesses are that these are unstable, expensive reagents. So TMT at present is 2,000 times more expensive than gold. The choice of mass spec is limited to those capable of measuring at these low MOV, especially if you want these 10 channels. And you require this Orbitrap fusion if you want these accurate ratios. Or if you're prepared to give the EASitag a try, maybe not. And you get lower coverage compared to label-free because the SPS-MS3 approach is quite slow. So is it possible to multiplex even higher? Yes, it is. You can actually combine CTAP or SILAC with TMT to do some really neat experiments. So I'm just going to point out these two papers, one from Chris Tape in Cell and one from Mikhail Savitsky, also in Cell recently. I encourage you to go and read those. So a label-free experiment then, which is the simplest of the options that you have available. In this case, all the processing and measurement steps are performed separately. Right? The comparison only occurs at the data analysis level. And this is frequently used in large-scale projects where you have more than a certain number of conditions or a certain number of patients that you want to measure, and you just don't have the patience to use SILAC spike-in for every single one of those. And the great thing about this is it gives rise to the simplest MS1 spectra relative to other strategies, and you get the best depth with this approach. So later on today, Hamid is going to give a detailed lecture about the MaxLFQ algorithm. So I'm not going to talk about that here. I'm just going to talk about the basics of label-free approaches, right? So there are in the early days, before quantitative information from mass spec was really taken into account, people used spectral counts as a measure of protein abundance. And so the mass spec operates in our case, we're always talking about data-dependent acquisition, and you essentially take the top n most abundant peaks, and this top, in doing so, each one of those n MS2 scans is called a spectral count. And the idea is that a more abundant protein will be sequenced more often, right? It will have more of those peaks in the top n of every MS1 scan. So you could then compare how many spectral counts you get from a protein in one sample to the same protein in another sample and deduce there was increased amount of this protein. Or you could go and just look at the extracted ion chromatogram of the peptides that gave rise to the intensity in your sample. So what this is the HPLC retention time, and this is intensity. So this is actually not continuous, but you have different MS1 scans that separate this and essentially give that you can convert into a peak. So initially, as your peptide elutes, it's low intense, and then you get to its peak, and then you get some tail here as well. And this extracted ion chromatogram is what we call an intensity, and this can be compared with that from a different sample. And this is much more quantitative. So this approach, it has 6 orders of magnitude for the difference in intensity between different samples, whereas spectral complexity is only 3 orders of magnitude. And spectral counting, rather, it's really bad at low abundant proteins. So as you get to low abundant proteins, the number of spectral counts that you will get for any given low abundant protein is quite random. So the MaxLFQ algorithm is an implementation of several normalization steps that overcome bias during sample preparation and MS measurement. It's not limited by multiplexing capabilities. It's very useful for clinical studies where you need to measure hundreds of samples and they don't fit into a TMT11 plex. And it will not work, though, if your samples are really too different. And a rule of thumb is if you try and process it with the MaxLFQ algorithm and it doesn't go through, then your samples are too different. But if it does go through, then it's good. It's not yet implemented for post-translational modifications. So when not to use? When would you not use LFQ? And the only answer there is when you have one of the other technologies has a specific advantage that you really want it for. So I know Matthias touched on this yesterday as well, but there are developments even in this approach. So this is this BoxCar strategy from Florian Meyer, and essentially the idea is that in the standard operation, the total signal of about 1 million ions or 1 million charges is concentrated within a few abundant peptides. So when you measure your MS1 scan, it's filled with these, all of the ions are essentially belonging to a few peaks, especially if you have a high dynamic range in the sample. So if you're measuring plasma, most of the ions will belong to peptides from albumin across the HPLC gradient. So what BoxCar does, it uses the quadrupole to distribute this charge capacity evenly over lots of narrow M/Z segments, allowing longer fill times for less abundant species. So now instead of having one MS1 scan, you have like 2 or 3 BoxCar scans where you basically change the quadrupole, allow accumulation of 100,000 ions in this small M/Z segment, and then change the quadrupole again and again and again, and you get 10 different small windows of the MS1 feature space, and then you inject all of those together, and essentially what you're doing is ironing out the differences in abundance between the different ions. And then MaxQuant has the capability to actually normalize this back. So I think at the moment it's definitely the only software that's capable of analyzing this kind of data. Has anyone ever tried to compare all of these different technologies? Yes, you will see a lot of papers in the literature that compare all of these different strategies. And my advice to you is, don't read them. Just if you understand the basic principles of the approaches that I've presented to you here, you can already tell yourself what strategy needs to be implemented, what features do I need most of all in my analysis. There are a couple of interesting papers maybe worth a read. There's this one by Jesper in Nature Communications benchmarking different quantification strategies for phosphoproteomics, and this one from Steve Gigi comparing TMT11 plex to an LFQ strategy. And I can summarize the paper for you here, that if you exactly had 11 samples, then TMT11 plex would be the perfect strategy to go for. If you have something that fits, if you're really interested in 3 conditions, right? Then you can fit all of those, all of your replicates into a single TMT 11-plex. You can fractionate your sample and get very deep in a reasonable amount of time, and you will get very good data. The problem comes when you want to measure more than 11 samples, right, or more than that number of replicates. And with the large-scale phosphoproteomics paper from Jesper, you can see that the idea of missing values that is frequent in phosphoproteomics can be overcome with the TMT approach as well. Again, limited by how many 11-plexes were compared in that experiment. So summary then of label-free approaches is that it's very good for large-scale projects. There's a high dynamic range, the best or deepest proteome coverage, certainly the least expensive. You've already spent $1 million on a mass spec. And then the weaknesses are there's the risk of systematic error due to parallel sample processing. The sample prep and the chromatography need to be very robust. It's time inefficient in terms of depth of coverage to a certain extent. If you want to get at the moment, and maybe Josh Kuhn has something to say about this on Wednesday with this FAMES device, which you can see in your abstract book, and also potentially the BoxCar as well. To get really deep coverage still requires fractionation, right? If you want to measure basically as many peptides as is possible to do, you need to fractionate, and that will require time. It has slightly lower precision than alternate methods, but is still the most accurate in one sense. And as I said, it can fail if you have insufficient common peptides between samples. That is quite a rare situation. It's only if you were comparing peptides from, say, a urine sample and a fecal sample, and I mean, from that, you would probably be publishing your research in a terrible journal. So the last point then is just to say that all of the things that I've described are possible to analyze in MaxQuant. So if you go, when we go to our session on, I can't remember what day it is now, if you go to our session where we do the MaxQuant stuff, you'll see that in this group-specific parameters, if you click on this standard, you'll see that all of these different ways to measure your sample are covered. So thank you very much, and that's the end.
We have time for a few questions. Please wait for Mike. Yeah.
Okay, so we start here. So hi, thank you for your talk. I was just wondering, for the isobaric labeling, do you see some issues with some natural occurring isotopes in the labels, or do you have a deviation?
So which isobaric labeling?
For TMT, for example.
For TMT, do you see issues?
So if you, I don't know, what's your thought on it? So if you're synthesizing your TMT labels, I can imagine there are some natural occurring isotopes in there, so they might deviate your mass as well. So is there any interference
So I know that the synthesis of these TMT reagents is not simple. So obviously the natural occurrence of heavy isotopes elsewhere will contribute on some level to heavy isotopes in the different channels. And when you order a batch of TMT, you actually get correction factors for how much how much heavy isotope is going to be in the neighboring channels.
The EASitag is very recent, so I don't know if you had practical experience with it. It's very interesting, but I wonder if the isolation window 0.4 requires the mass spectrometer to be in perfect shape all the time, and if that is going to change the routine operation. I mean, I expect it to be a more instrument demanding label.
Yeah, I would. So I don't have practical experience of running the experiments myself. So obviously I work closely with the guys who developed it, and I don't think they've had to clean their quadrupole any more frequently than they do at the moment. So the downside would be if your quadrupole is dirty, then you would get poor transmission efficiency, right? So every time you tried to collect 100,000 ions, it would just take longer. But when you do collect them and fragment them, your quantification is not going to be affected. Okay, last at the back. There.
And is the EASitag going to be commercialized like TMT and, let's say, financially as demanding?
As financially demanding, I can't say, but it certainly will be commercialized. Yeah, these things take time, and I'm not sure how soon it will be on the market. If you were really keen on getting it sorted, then I'm sure that if you send an email to Matthias or actually the senior author on that paper is Felix Meissner. He was the guy who co-developed the chemistry of the tag. If you email him and say that you're really interested, then maybe they could even give you some. Okay. Oh. We're running out of time now.
So, hello. Regarding the label-free quantification, I heard that the transproteomic pipeline introduced something called spectral index counting. Okay. It's like a hybrid between measuring the intensity of a peptide with spectral counting to perform label-free quantification. And they say that it should be much faster and much more robust than just spectral counting. So my question is, have you tried it out, and do you know if it's as promising as they say?
I mean, I think this. I have to say, I'm not aware of this, but I would be surprised if there were any form of data analysis that could overcome this limitation with spectral counting, like the orders of magnitude it's able to measure. But yeah, maybe we can talk about that. And you can. Yeah. Yeah. Thank you.